For chapter 2, we're going to focus on cells, the structures of cells, as well as the organelles that are found within cells. And we're going to look at how that determines their function. Before we do that, we're going to start with cell theory. Cell theory says that a cell is a structural and functional unit of life. So that means that all living organisms have cells and are made up of cells. The structure and the function are going to be complementary for these cells. And again, that's going to mean that the structure that the cell has or the shape that the cell has is going to dictate what kind of function it can have. And then the continuity of life has a cellular basis. That's because cells come from pre-existing cells. They don't generate out of nothing. In the body, there's trillions of cells, and out of these trillions of cells, there's 250 different types of cells in the human body. All of these cells are going to differ in their size, shape, organelles that they have, and these differences are going to lead to differences in functions. So here we can see some of the diversity that you can find. So for example, these epithelial cells, these are the cells that make up the epithelial tissue, which mainly function in covering surfaces or lining the, the hollow organs, the inside of hollow organs, I should say. There's erythrocytes, which are red blood cells. They mainly function as, carrying oxygen and carrying nutrients within the bloodstream. You have muscle cells, which make up muscle tissue. So it can either be smooth muscle, skeletal muscle, or cardiac muscle. The main function for these is to generate movement. We have immune cells, like a macrophage that you can see here, which mainly function in the immune system. Adipocytes mainly function in storing nutrients. We have nerve cells, which are neurons, mainly function in communication because they send signals uh, from one cell to another. Then we have reproductive cells like the sperm cell or the oocyte. When we look at the subcellular components of cells, we're going to look at the organelles. So, so this here is just a general model of a cell where you can see the different types of organelles that the cell can have. But just realize that depending on the cell type, that'll determine what kind of organelles are more abundant. So if it's a cell that has a high metabolic rate that uses a lot of oxygen, you're going to see that that cell type would have more mitochondria. So for example, in muscle cells or cells that are found within the kidney, renal cells, because they are highly metabolic and use a lot of oxygen, those types of cells have a lot of mitochondria within them relative to other cells. If the cell's involved in forming a lot of proteins, it's going to have a lot of ribosomes within them because ribosomes are organelles that synthesize proteins. So here, when we look at a generalized cell, we're going to see the common structures and functions of these organelles. We're going to start with the outer barrier of the cell, which is a plasma membrane. So this is the outer barrier that separates the intracellular fluid from the extracellular fluid. So fluid found on the outside versus fluid found on the inside. That's separated by this cell membrane, also known as the plasma membrane. Then we have the cytoplasm of the cell, which includes the fluid portion of the inside of the cell, and also the organelles that are found within the fluid. And then we have the nucleus of the cell. This is where a lot of the genetic information is housed. Well, I should say where the genetic information is housed. It's housed in here, the nucleus. Okay. So here you can get a closer look at the organelles. The biggest organelle is going to be the nucleus. It's usually very obvious when you look at a cell, whether you're looking at it on a tissue slide. Although some cells do have do not have a nucleus. They're considered to be anucleate. But for the most part, when you look at a cell, you're going to be able to see the nucleus. And then we can see other components of the cells like the mitochondria here, which is the one that's involved in generating ATP. We have the rough ER and smooth ER. So rough endoplasmic reticulum and smooth endoplasmic reticulum. We have things like the the ribosome. Ribosomes are usually found within the cell and also on the rough ER. So that's what gives the rough ER its name, because on the membrane of these little sacs, you have ribosomes attached to it. So it gives it a rougher appearance compared to the smooth ER. Okay. So we focus on the plasma membrane. So if we zoom into this outer boundary here of the cell, we're going to see that it acts as a barrier. So it does separate the intracellular fluid from the extracellular fluid. It plays a role in cell activity because it controls what can come into the cell and also what can leave the cell. So it makes a semi-permeable membrane. Semi meaning uh, partial, and permeable obviously means the ability for something to cross. So only some substances are able to cross the semi-permeable membrane. So this can be a benefit or a detriment. Obviously, the benefit would mean that it prevents things that are harmful to the cell from penetrating the cell, but it also means that things that the cell needs might also be inhibited. So it might also prevent those items from coming into the cell. To work around that, there are certain structures on the cell membrane that allow for certain things to come in. But those are just some examples. So this semi-permeable membrane, it plays a role in the cell activity by controlling what comes in and out. But this membrane is in constant flux. So it's also known as a fluid mosaic because the membrane proteins that are found within the cell membrane are constantly moving. Inside of or within the cell membrane, we can see that it's going to consist of different structures like membrane lipids. So these are going to form the lipid bilayer. We have specialized membrane proteins, which are going to float through this fluid membrane, which again gives it that constant fluctuating pattern that's known as the fluid mosaic. We're also going to see sugars, which are referred to as glycocalyx. So these are carbohydrates that attach to cell membrane structures. Within the cell membrane, there's also membrane structures that help hold the cells together through cell junctions. So these could be things like uh, structures that are known as desmosomes, which prevent the cells from being pulled apart. So in certain areas where there's a lot of mobility, a lot of wear and tear, like cardiac tissue, because the heart is constantly contracting, the cells that make up the heart are in constant motion. So to prevent those cells from coming apart, there's cell junctions which hold those cells together. But here we're going to zoom into the cell membrane to see the different components. So here's the phospholipid bilayer. So if you look closely, you can see that this is made up of two layers. Okay? That's why it's known as a bilayer, because it's two layers. These are going to be made up of these little structures that have a hydrophobic tail and a hydrophilic head. So that means that one side is going to be it's going to like to interact with water or fluid, and the other side is going to not want. It's going to dislike interacting with water or fluid. So that side is always facing each other, and the heads are always facing towards the fluid. Embedded within the membrane, there's also these cholesterol components, which give it a lot of its makes the cell membrane. It gives it a more of a structural support for the cell membrane, and it also helps prevent things from penetrating the cell. So anything that's hydrophilic would not be able to cross the lipid bilayer because of the components like the cholesterol, which prevent those hydrophilic molecules from crossing through. Then there's these proteins, which have a lot of diversity among them, because the proteins can either be channels that allow for things to go from the outside of the cell into the cell, or vice versa, things from inside of the cell to leave the cell. And then there's also these carbohydrates, which are which form the glycocalyx. So these can attach to either lipids, like it's shown here, or to the proteins. And these glycocalyx mainly serve as identity markers. So it allows the cells to recognize each other. For example, it'll allow the immune cells to recognize its, cells from within the body so that it doesn't attack them. It also helps to sort things um, by allowing certain tissue cells to aggregate together. For the lipid bilayer, here you can see the components of it. You don't have to really memorize these percentages. It's just to give you an idea of how they're the abundance of each type. But it's mainly made up of the phospholipids, which are these lipids here. Okay. As you can see, they do make up most of the membrane. So it's mainly phospholipids, which have the two parts. The phosphate heads, which are polar meaning that they're charged, and these are the water-loving parts. So these are known to be hydrophilic. The fatty acid cells, these are the nonpolar parts because they have no charge, and these are the water-hating components. So these are hydrophobic. Then there's the glycolipids, which are, the least abundant, but these are lipids with sugar groups on the outer membrane surface. So it would be like these here. So the sugar groups are represented by these green structures. If the sugar group attaches to a lipid, it forms a glycolipid. And then there's the cholesterol, which again is just there to increase the stability of the cell membrane. This is how the phospholipids kind of organize. They always want the heads always want to face the fluid portion. So whether it's the intracellular fluid or the extracellular fluid, and the tails always face away from it. That forms the lipid bilayer. For proteins, there's integral proteins, which means that they're firmly embedded in the lipid bilayer or in the cell membrane, or they're going to be attached to the lipid bilayer. Uh, they're going to form short chains with carbohydrates. So that's going to be known as a glycocalyx. So if you have a sugar group that attaches to a protein, or if you have a sugar group that attaches to the lipid, that sugar group is known as the glycocalyx. Okay. And then we have the peripheral proteins, which mainly are going to be seen on the membrane surface. So these are here to support the plasma membrane from the from uh, from the cytoplasmic side, or so this would be the peripheral proteins. So you can either find them on either side, on the cytoplasmic side or the extracellular fluid side. These would be the integral proteins because these are the ones that are embedded within the cell membrane. If we focus on these integral proteins, we can see that they can have different functions. So they are able to perform many different tasks like transport because they can be in the form of channels. So a channel will allow things to enter the cell or it'll allow things to exit the cell. Okay. Then we have receptors for signal transduction. So that means that the protein itself will be the receptor, and then it'll allow for a messenger to attach to the receptor. So and that messenger can be either a neurotransmitter or a hormone. And when the that messenger attaches to the protein, the protein changes shape, and then that will also allow for things to either enter the cell or exit the cell. Other proteins that are found within the cell membrane can act as enzymes. So these are proteins that are activated, and when they are activated, that also it changes the protein's shape, and that can lead to different things. So it can either allow for things to enter or exit the cell as well. There's cell-to-cell recognition. So these are the glycoproteins that attach to the sugar group, which then forms the glycocalyx. And these are mainly there to serve as identity markers to be recognized by other cells. Other functions that the proteins can have, so they can either serve as attachment sites for the cytoskeleton. Cytoskeleton is like the skeleton of the cell. It's mainly made up of filaments. So they can act as attachment sites for these filaments, or they can also serve as attachment sites for fibers that are found in the extracellular matrix. So a lot of the protein fibers, like collagen, uh, can attach to these uh, to these proteins on the cell membrane. And then cell-to-cell joining, that would be these. You can see these in areas where there's a lot of mobility in the tissue. So areas where the organ is constantly moving, and so that means that the tissue is constantly moving. And so to prevent those tissues from coming apart just through wear and tear, you have these cell-to-cell joinings, which hold the cells together. So I mentioned this at the beginning where it would be like cardiac tissue. There's a lot of cell-to-cell joinings, so in the form of desmosomes that the it helps keep the cells locked in. Here we're going to see how the different types of vesicular transport. So this involves the transport of large particles, or it can be macromolecules and fluids that cross the cell membrane through these little sacs that are called vesicles. So if it's through exocytosis, that means that the components are going to these are going to be things that leave the cell. So they go from inside of the cell and they get taken out. Endocytosis, that's when the cell brings things in. And as far as for endocytosis, the one that you have to be familiar with is phagocytosis. So this is where a large uh, molecule or like a large particle is engulfed by the cell, okay, and then taken into the cell. But there's other different types like penocytosis. This would be mainly for fluid, and then receptor-mediated endocytosis. That means that in order for the cell to bring something in, it would have to be there has to be a receptor that's activated first. And then exocytosis, that just means that it's taking the components out of the cell. So it forms the vesicles within the cell, and then that vesicle will travel to the cell membrane, and there the contents will be released. Here, you look at this. Here, just to show you, give you an image of what it looks like when the cell is using phagocytosis, how it's taking something from the extracellular matrix into the cell itself. And this yellow component would be the cytoplasm. So this is the inside of the cell. This thin black line, that's the cell membrane. And then this blank area, that's the extracellular fluid. So that's the outside of the cell. For the fluid portion of cell, that is part of the cytoplasm. So the cytoplasm is made up of the cytosol. That's the gel-like solution. It's made up of mainly water and other soluble molecules like proteins or sugars. There's also inclusions, which are insoluble molecules that are also found within the cell. So there could be things like glycogen, which are polysaccharides. So it's a more complex form of glucose. You have granules and pigments, as well as things like vacuoles and crystals. Then you have the organelles that are found within the cytoplasm, and so these are the metabolic structures of the cell, uh, each one giving the cell a specific function. So so if you're including the organelles and inclusions, that fluid portion of the cell is considered the cytoplasm. But if you're just referring to the fluid itself without including the organelles and inclusions, that's called the cytosol. So that's the difference here. Within the cell, you're going to see that there's different types of organelles, and they're classified into two categories. There's the membranous organelles, which have a plasma membrane, and then there's the non-membranous, which do not have a plasma membrane. We're going to start with the mitochondria, which is known as the power plant of cells. It's known as a power plant because this is where ATP is generated. Within the mitochondria, you're going to see uh, structures that are called cristae, and these are just membranes that are found within the mitochondria uh, that are folded in a very compressed position. Within mitochondria, you're going to see that it contains their own DNA as well as RNA and ribosomes. Mitochondria, they mainly resemble bacteria. So it is thought that the way that the mitochondria found its way into the cell was because millions and millions of years ago there was a prokaryotic cell that was engulfed by a eukaryotic cell, and then they formed the symbiotic relationship, and that relationship eventually turned into what you see now, where you have an organelle like the mitochondria that has its own separate DNA than the cell that it's housed in. Okay? So the DNA in the mitochondria is different than the DNA that's found in the nucleus of a cell. Ribosomes, these are the organelles that synthesize proteins. They're made up of proteins and ribosomal RNA. And there's two different forms that are found. So there's free ribosomes that are free floating. These are going to synthesize proteins that are going to go to they're going to go to organelles within the cytoplasm. Then there's membrane-bound ribosomes that attach to the membrane of the endoplasmic reticulum. These are going to synthesize proteins that are going to be incorporated into the cell membrane. Endoplasmic reticulum, that's another uh, organelle that it's found within the cell. So this is going to be found in two varieties. There's rough ER and smooth ER. And just realize that the ER is continuous with the outer nuclear membrane. Okay, so the nuclear membrane would be the membrane that's found uh, on the nucleus. So it's the outer boundary of the nucleus. The rough ER, this is going to appear rough because this is the endoplasmic reticulum that has proteins on the surface of it. So because of the presence of those, well, sorry, not proteins, ribosomes, that gives it the rough appearance. They're mainly involved in synthesizing proteins here. The proteins that are synthesized are going to be enclosed in vesicles, and these vesicles, these little packets of proteins, are going to be sent to the Golgi apparatus where they're processed. So within the rough ER, you have these uh, ribosomes that are found throughout the rough ER. It gives it the rough appearance. Ribosomes are the ones that synthesize proteins. So when they do synthesize them, when they make them, they're going to be packaged into the vesicles, and then the vesicles are going to find their way from one side of the rough ER to the other, where they're going to meet the Golgi apparatus. Then there's a smooth ER. So these do not have the ribosomes, which gives it a more smoother appearance. They're mainly involved in lipid metabolism, absorption and synthesis of fats. They're also involved in detoxification of chemicals, as well as converting glycogen to glucose, because glycogen is the more complex form of glucose. This is considered to be a polysaccharide. Glucose is considered to be a monosaccharide. Smooth ER is also involved in the storage and release of calcium. But moving on to the Golgi apparatus. So Golgi apparatus is known as the post office of the cell because it does a lot of the things that the post office does. So it modifies, it concentrates, and it distributes packages, okay, similar to what the post office does. When it receives a mail or a package, it'll apply a label to that package, and then it'll sort it to different areas. And then from those areas, it is delivered to its designation. Because remember that the proteins are made here at the rough ER, because this is the endoplasmic reticulum that has the ribosomes. So proteins are synthesized in the ribosomes. Once they're synthesized, they're going to be packaged into vesicles. So these vesicles are then going to find their way from one side of the rough ER to the other. When they find their way to the more exterior region of the rough ER, it's going to meet the Golgi apparatus. The Golgi apparatus is going to receive those vesicles, and then when it receives them, it's going to modify them by adding and removing the the sugar components. Okay, so it's going to add like the glycocalyx labels. So it adds them and removes them. That's the modification that occurs. Depending on how they're modified, that's going to determine what fate it has. So what those proteins are eventually going to be turned into, because they can either be modified to exit the cell. So in that case, the vesicle is just going to find its way to the cell membrane, and then it'll eject the contents into the extracellular fluid. Or they're going to be modified to be incorporated into the cell. So this is where the proteins just find their way to the cell membrane, but then they're just incorporated into the cell membrane as either peripheral proteins or integral proteins. Then the other fate that they can have is to be turned into lysosomes or into the components of lysosomes. So you can see here that this vesicle is destined to be a lysosome. Okay. Peroxisomes, these are membranous sacs that contain powerful detoxifying substances that neutralize toxins. So free radicals. Free radicals are toxins that are natural byproducts of metabolism. So they just naturally build up um, in your body. The two main detoxifiers are oxidase. So these are enzymes, oxidase and catalase. Oxidase uses oxygen to convert those toxins into hydrogen peroxide. Okay, so you're probably familiar with hydrogen peroxide. So this is naturally produced in your body, but also hydrogen peroxide is also a toxic substance in itself. But the peroxisomes also contain another enzyme that's called catalase, and that turns the hydrogen peroxide into water. Peroxisomes also play a role in the breakdown and the synthesis of fatty acids, so they're also involved in lipid metabolism. Lysosomes, it's another type of organelle. These are more spherical membranous bags that contain digestive enzymes within them. Their main function is to ingest bacteria or viruses or other toxins. They they degrade non-functional organelles. So if the cell if the cell has a lot of organelles that are non-functional, then those lysosomes are going to break those organelles down. Because you can't have the cell with non-functional organelles, because then the cell is no longer able to perform its function, and that could be detrimental to the organism. Um, Lysosomes are also involved in metabolic functions like the breakdown of glycogen into glucose, also the breakdown and release of calcium from the bone, because your bone stores a lot of your calcium, and when the body needs to elevate calcium levels, it needs to break it down and needs to release it from the bone where it's stored. So lysosomes are involved in that. And then they're also involved in the intracellular release of these substances so that the injured cell uh, eventually digests itself. It'll digest itself because when the lysosome releases its contents, those those digestive enzymes are going to be released within the cell, and the cell will digest itself. The cytoskeleton is a network of rods that run through the cytosol. Remember that the cytosol is just the fluid portion of the cell. Okay? If you include the organelles with the fluid portion of the cell, that's known as a cytoplasm. So it's just to clarify the terminology here. So there's hundreds of different types of proteins that link rods to other structures. So they mainly act as the skeleton of the cell. And there's three different types. There's the microfilaments, which you can see here. There's the intermediate filaments, and there's also the microtubules. So they're mainly there to give the cell its form. It gives the cell its shape. The nucleus, this is the largest organelle that's found within the cell. Usually you're able to see the nucleus when you look at a tissue slide, although there are some cells that are anucleate. Um, well, actually, yeah, I jumped ahead a little here, but most cells do have a nucleus, so they're considered uninucleate or mononucleate. Skeletal muscle cells, these are uh, specific examples of cells that have many nuclei, so they're known to be multinucleate. And that's because during embryonic development, they do start off as individual cells, but they do merge, so to speak. And so, because of the merging that occurs, it retains the nuclei of those cells that merge. So the final product does have multiple nuclei. And then there's cells that are anucleate, like the red blood cells. So they do not have a nucleus within them. And that's mainly because of their function. The main function for the red blood cells is to carry things, to carry oxygen, carbon dioxide, or nutrients. So it has no need for the nucleus because the nucleus is mainly there it mainly has the genetic information that's needed to produce proteins. But going back to our objective for this lecture, the structure of a cell or the structure of an organelle determines what kind of function it can have. For the structure of the nucleus, if we if we take a closer look at it, we can see that the nucleus has three main structures. There's the nuclear envelope, which is a double membrane barrier, and there's two of them, right? Double membrane. Outer is continuous with roughly are, and it's also embedded with ribosomes. The inner is the one that's deep to that, and this is it's mainly there to give the, it maintains the form of the nucleus. This is the part that adds as a scaffolding for DNA, because DNA is found within the ribosome in the condensed form, and it does attach to the inner membrane. Okay. And there's also the nucleoli, which is the dark staining component of the DNA. This is where the ribosomes are assembled. So the small ribosome components and the large ribosome components are put together. Chromatin is a condensed form of DNA. It's also found throughout or within the nucleus. Chromatin is similar to chromosome, but chromosome would be a more condensed form of chromatin. That's only found when the cell is replicating. But if it's not replicating, then the DNA is in the chromatin form. Okay, so that's going to be it for this chapter, and we'll continue on the next chapter in person.